So yeah, my name is Anke Schulte. I'm Ms. Bayer. I'm in CGT Research Early Development. I'm here to introduce Lucas Harrington. You heard his name already before in the presentation by Florian. So Lucas Harrington is the CSO of Mammoth Bioscience and the co-founder of Mammoth Bioscience. So he studied biology in University in St. Louis. Some of you will know, because crop science is there also. And then he moved on and did his PhD in the laboratory of the University of California in Berkeley. And he did his PhD in the laboratory of Jennifer Doudna. Jennifer Doudna will be known to you. She received the Nobel Prize for CRISPR-Cas in 2020 together with Emmanuel Capetier. So since then, Lucas moved on, and he's, as I said, Chief Scientific Officer of the company Mammoth Bioscience and the co-founder of this company, where Jennifer Doudna is the head of the Scientific Advisory Board. So we as Bayer are in partnership with Mammoth Bioscience since a year ago. It's pretty much not close, it's 10 months now. And why did we do this? Because Mammoth Bioscience is a company who's focusing on the new way of finding, on the second generation of Cas, acting in a different way, cleaving in a different way, and in particular also being smaller than Cas9, which Florian alluded to. So I don't want to say anything more about this. I think Lucas will give us a very detailed explanation and dive with us together in the activities they have running at Mammoth Bioscience. And afterwards, there will be time for question and answers in this round. So I would like to hand over the floor to Lucas. Lucas, where are you?
Over here, Anke.
There you are.
All right. All right. Thank you, Anke, for the very kind introduction, and thanks, Florian, for the introduction generally to CRISPR. Yeah, hopefully I'll be able to glance over some of the kind of general things, and we can get into the interesting stuff. So really, what the foundation of the company Mammoth Biosciences is how we can harness the huge natural diversity of CRISPR systems to actually build better systems beyond the legacy systems like Cas9. So if there's just one thing that you take away from the talk today, I think I want it to be that we're really at just the tip of the iceberg in terms of where we're at with CRISPR. It's moved at a breakneck pace. Yeah, you saw how many different programs are in the clinic, but the potential when we're able to just reprogram biology essentially however we want is pretty endless. And so really what we look to is what's the next decade of CRISPR look like, and recognizing that the work that Jennifer and Emmanuel did back in 2012 was really just the start. So we're going to cover a lot of different topics today. Mammoth is a company that has a diverse portfolio of different activities. I'm first going to dig in a little bit more to what Florian touched on, which is just the diversity of CRISPR overall, starting with how these systems function naturally and how that's led to the diversity that's out there. I'll then get into our ultra-compact nucleases and talk about why those are important, but also some of the work that has to go in to actually take something from a discovery to actually getting it to work as something that has therapeutic potential. And then I'll talk about CRISPR not for direct therapeutic applications, but actually for disease detection. So the same function of being able to reprogram it with a guide is actually incredibly powerful for decentralized diagnostics as well, which when we think about genomic medicine, it's not just how do you cure the patient or how do you treat the patient, but it's also how do you identify the patient and monitor them. And of course, this has applications beyond human health as well. So we'll get into it. So Florian touched on this, but coming from the Doudna lab, we kind of have to talk about how really CRISPR is kind of this gift from basic biology of just trying to understand how does nature naturally work to protect itself when you're talking about bacteria that get viruses just like we do, bacteriophage in this case. And this is a extremely elegant system that people observed nearly 2 decades ago or 3 decades ago now, and it really wasn't until the last decade that we really started to understand how does this work. So essentially, what happens with CRISPR in its natural environment is you have what's called the acquisition stage, where fragments of foreign DNA are able to be acquired and put into the genome. Those then program the systems like Cas9, and then those are able to go and target those invading bacteriophage. And the reason this is important is that, again, Florian mentioned this, but there's tremendous pressure here to diversify and to have a number of different activities. And that's really what's led to this explosion, is just this constant arms race between the pathogen and its host. And one of the big things that we recognized, this is going back to for CRISPR timelines, ancient history back in 2014 or 2015, one thing we started to recognize is that a lot of the systems that we were using back then were just systems from isolated bacteria, so things that we could grow on a plate. Of course, over the last decade, we've had this explosion of effort in metagenomics, so actually trying to understand the microbial universe that can't be cultivated, and using, of course, the advances that's been made in NGS to be able to study and characterize that. And so, the insight that we had was to actually look into this, 99% of microbes that are not able to be cultivated and see what their CRISPR systems and their adaptive immune systems look like. And so this is really what's led to this explosion of different CRISPR systems. So back in 2012, really all we had was Cas9, and this is kind of where we got our start looking at metagenomics. Looking at just new different versions of Cas9 that have different activities, focused on things like thermophilic Cas9s. But really, what's happened over the last 10 years now is this huge explosion, and this is extremely out of date actually, and there's quite a few more beyond this, but a huge explosion of different CRISPR systems, and in particular, there's a lot of different features in these CRISPR systems that you can leverage. So, when we think about Cas9, that really was just the first system that people got to be reconstituted, and it works well enough. You saw, the advancement that it's had in the clinic, but there's no reason to think that, of the hundreds of thousands of CRISPR systems out there, that's actually the best one. It's really just kind of momentum and an artifact of history of Jennifer getting that system to work and Martin getting that system to work. So what we've done at Mammoth is to really scale up this process of looking through that diversity. And there's hundreds of thousands of these systems, so it's not trivial to try and find that needle in the haystack, the systems that are going to perform best. And so we have, of course, a big focus on automation and high-throughput screening and the assays that we use to understand those CRISPR systems and how they work. But we of course, take those. We then characterize them. Each CRISPR system, we get kind of a spec sheet of what its features are. And I think an important thing to note, too, is Florian, if we take Cas14 as an example, Cas14 is not a single CRISPR system. It's actually, there's probably tens of thousands of variants of Cas14 alone. And so when we talk about understanding this diversity, it's not just finding new named CRISPR systems, but it's also finding the best systems within those different subsets of systems. So, we take this process, we go and screen through the different CRISPR systems, and from there, we are able to push them into different applications. And I think a key fundamental thing that we focus on as well is that there's not going to be a one-size-fits-all CRISPR solution, right? There's going to be, different requirements that you have for different tasks, and so you do need this portfolio of enzymes. The other thing I'll mention, because I think it's important to actually getting systems that work well, is we use this natural diversity to kind of make leaps and to explore hugely different areas where often less than 10% of the protein is conserved. But then protein engineering is also, of course, a huge process and effort where you're able to hone features and actually do things that nature might not have the pressure to evolve. So 2 examples that we're going to take through today of how this diversity is used. One is on these ultra-compact systems. So we'll focus on Cas-V. I'll mention this newer system that we have as well briefly. And then we'll talk about CRISPR for disease detection and specifically how you can drive CRISPR and diagnostics into a decentralized setting. All right, so to get into ultra-compact systems. So Florian touched on this, but CRISPR is really fundamentally a different type of therapy than what we're used to, right? When you think about small molecules or siRNAs, ASOs, these are transient methods where you're really trying to maintain the patient's well-being, and often you have problems of just patient compliance. You're talking about therapies that do not have full therapeutic outcomes in terms of actually being able to preserve the natural state. And of course, if you're talking about something like an ASO, you may have to actually do intrathecal injection every couple months, which of course is a huge patient burden. And really, what CRISPR is offering is this one-time curative model. So it's a very different way of thinking about medicine and healthcare than what we're used to, especially what we're used to in the U.S. for sure. I think this is something that's got a lot of potential. A lot of the therapies that have been out there so far, as Florian mentioned, it's really been focused on ex vivo therapies. And I think this is a great proof of concept and demonstration of the CRISPR tools working. But really, that's not what the promise is. It's being able to do a simple, IV injection or one-time treatment where that patient is then, fully restored and no longer is even considered a patient besides, routine monitoring. The biggest problem with doing these in vivo therapies, and the reason that you see all of the programs focused on ex vivo as well as hepatic targeting in the liver, is delivery, right? It's how do you actually get these systems into the proper cells to have the editing outcome that you need. There's really two delivery tools that we have. There's lots of interesting research going on new delivery tools, but really there's either LNPs or AAVs. LNPs largely restricted to the liver currently, but really AAVs, there's been a tremendous amount of work over the last 1996 was the first clinical trial for AAVs. There's been a lot of work to understand these and a ton of new capsids that are being developed to capture other tissues beyond the ones that natural serotypes target. And the big challenge, of course, with AAVs, there's some safety challenges, which I think a lot of these new capsids are starting to address. But a lot of it is the cargo capacity. So you have about a 5 kb capacity for a standard AAV, and when you think about Cas9, these systems just they're too big to be packaged into a typical AAV. And so, what we did, and we started to see these systems naturally, is these extra small systems. So it's good to kind of put some reference there. But we are talking about systems that are right around 400 to 450 amino acids. So about a third of the size of those legacy systems like Cas9. This opens up a lot of tissue-specific work that you can do besides hepatic targeting, and really able to deliver on much more of the promise of CRISPR and genetic medicine. So to talk about a few of the systems that we're going to touch on today, down here for reference, we have some of the legacy systems like Cas9 and Cas12a, and these bars are meant to represent the size. With small variants, or Cas-X, which is a system that we discovered back in Berkeley, which was kind of the small system of the day. These are around 1,000 amino acids, around 950 at the smaller end, but still relatively large systems. Systems that we're talking about here, Cas9, which we'll focus on today, is right around 700 amino acids, and then Cas14 and a newer system called NanoCas, which isn't on here, which are right around 400 or 450 amino acids. So if we go back to that previous slide, you're talking about systems now that really only occupy about half of the AAV. And so all this extra cargo room can be used for those CRISPR plus fusions like we mentioned. So this would be things like base editing or gene writing, multiple guide RNAs, tissue-specific regulatory elements, but you're really able to have a lot more flexibility with the therapies that you're building using these small systems. And one of the cool, I think this is a good example to highlight of why it's important to understand this natural diversity, is the system Cas14. This is true of a number of other systems as well. But a lot of time, so when we originally discovered these back in 2016, a lot of people were looking at them and just said, these are too small. They're not going to work. They clearly don't. They're missing something. They don't have the machinery that's required. And after investigating them and starting to really look into it, and this is a structure that was published a few years ago, what we found was that these Cas14 proteins actually form a homodimer. So these are the same proteins here, just shown in different colors, and they adopt slightly different folds. So it's an asymmetric homodimer, and these scaffold onto a guide RNA to make a much larger CRISPR system. So if you were going to try and make a smaller CRISPR system through protein engineering, however smart you are, it'd be take forever or not really be possible to try and get a system that has this elegant of a solution for how efficiently it can be encoded. So a good example of why we need this balance of exploring natural diversity as well as protein engineering. So today we're going to focus on Cas-Phi, though, which is this kind of, for us, a medium-sized system. For other people, maybe ultra-compact. It's got a number of different interesting features. It's found in large bacteriophage. So again, another example of why metagenomics. These are not phage that you could grow on a plate, but these are almost exclusively found in those sources. Another cool feature that we'll touch on in a little bit is how it processes its own guide. So again, another example of how nature provides new features for these CRISPR systems. So this is an example of what it actually takes, and there's probably another 50 orthologs that were screened that aren't shown here just for simplicity. But each of these dots is a guide, a different guide RNA tested in triplicate. This was the original published Cas-Phi over here on your left. And you can see it's working reasonably, moderately, I'd say, for genome editing here. And just through screening these natural diversity, so this isn't doing any engineering or anything like that. We're able to get systems that work about 10 times better just through looking at that natural diversity. So I think it's unclear to us what makes a system exceptional, but really what it requires, and of course as a scientist I wish this wasn't the case, but it requires just brute force to look at every system out there to get the best system possible. Just to highlight another example of how we now take that system that works much better than the original variants and do protein engineering. So this is a scanning mutagenesis experiment. Typically these are done sequentially and you cycle them over and over. But here we're just scanning positively charged residues across the protein sequence and seeing what the effect is on indel formation across two different targets here, target 1 and target 2. And there's this clear exceptional mutant here, again, just showing one example of this for simplicity, but a system that works typically 2 or 3 times better than the wild type variant. And in terms of off-target profiling, the system seems to be extremely accurate as well. So again, a simple point mutation can make a system that is working okay to something that's working much, much better without too much engineering effort and with a relatively simple library design. So, we won't go into too much detail on the different processes that we run it through, but again, just showing you kind of what the general process is for taking something that you see in nature and getting it to work more robustly and getting it to work at therapeutically relevant levels. So now I'll talk about some of the different applications that we can do with this. Given the focus on on in vivo, I won't touch on this too much, but really just to show that we can do what you can do with Cas9. So this is editing T cells, a relatively kind of routine CD19 CAR engineering using a knock-in. And you can see, even with triple knockouts, we're able to get very good editing efficiencies with this Cas-Phi system, despite it being relatively young compared to the legacy Cas9 systems. Across a variety of different cell types, and this is again just a small sample. You can see it's working well. So this is in cancer cell lines. You can see getting about 99% almost complete editing there. This is in mouse hepatocytes. Again, very robust editing, about 80%. And here, looking at PCSK9 levels, so a typical target that we'll use, you can see almost complete reduction there. And then over here, iPSC cells editing, so again, at about 80%. So again, the system works robustly in a variety of different ex vivo applications, but really what the point is to use this in vivo. And so here's some, one of the early mouse data studies that we generated with Cas-Phi. So here, this is a simple single AAV8 injection, tail vein injection, and we're targeting PCSK9. PCSK9 is a target that is kind of ubiquitous in the CRISPR fields at this point. We're not particularly interested in it as a therapeutic target, but it's the target also that Verve has been pushing forward with base editing. And the idea here is it's an easy target because you can just look at cholesterol in the blood as well as PCSK9 serum levels. So it's a good kind of proof of concept target to go after. So here we're comparing the legacy Cas9 system, again, going through about a decade of optimization with this system versus the relatively new Cas-Phi system. You can see comparable indels between those two. And importantly, in terms of the actual protein levels for PCSK9, you can see a near complete reduction for both of those proteins. So, again, an example where, it's a new system, but we're able to get very robust indel generation with these new proteins. And then I guess here I'm going to kind of jump into a few additional features with this system. I think this is kind of the fun part where when you get a new CRISPR system, you get it to work, and of course you need to show efficacy. But then as you start to investigate it, you start to pull out other features that these systems have that actually open up new applications. And I think it's kind of like a Christmas morning when you start to do the biochemistry and understand what these systems are able to do. So what I'm showing here is basically, we often talk about fidelity as a safety risk. Here I'm actually showing fidelity as a feature of the enzyme. So we have two targets. I've obscured the specific genetic target here, but it's a natural genetic mutation that varies just by a single SNP. And then we have this Cas-Phi enzyme that's targeted either towards one or the other. Or it's the same guide here, and the wild type versus the mutant target. So starting with Cas9, you can see very little discrimination. So if both of these lines are high, that's not good. That means it's not able to discriminate between the two targets. We're getting across a dose titration here, robust editing against both of those targets despite the mutation being there. Cas-Phi, on the other hand, again, a dose titration on the bottom. What you can see is still getting robust editing for the on-target mutation, or on-target wild-type target, but the point mutation is able to completely ablate the editing of the system. And so this has been, kind of something that people have hoped that you'd be able to do with Cas9 for some time now with this allele-specific targeting, and have done a lot of kind of maybe convoluted tricks to try and get Cas9 to be able to do this, but just out of the box, really across the spacer sequence, you're able to do this with Cas-Phi. So another cool feature of this system is that it, like I mentioned, it can process its own guide. And so this is actually a nice feature for multiplexing where you want to edit multiple targets simultaneously. So typically, if you wanted to do that, you'd need to feed in 3 different guide RNAs or however many guide RNAs you want, and have different promoters there or put ribozymes in between them. What this system can do is if you feed in what we call a CRISPR array, so this is kind of how they naturally look actually in the bacteria. So R being repeat and S being spacer, so the constant region versus the variable target. You can concatenate those together, and the Cas-Phi enzyme, after it's expressed, will actually go and parse those up into individual guide sequences, and then those molecules can go target independently in the genome. And so what you can see here is, and this is normalized to the relative activity of the individual guide RNA, since there's of course some variability between the just efficiency of each guide independently. What you can see here is that across these spacers, when we feed them in a string, you can get robust editing at all 3 sites. Typically, we do see a little bit of a deficit at spacer 1, but if you continue this out to spacer 4, 5, 6, you'll also see robust editing there. So a very simple way to edit multiple targets without having to get the complex vector design that you'd need to do for a Cas9 system. All right, and so one of the big reasons we care about these ultra-compact systems is because the toolbox for editing has really expanded pretty dramatically over the last decade. So for things like gene writing, epigenetic editing is of course an area that's growing in popularity as well. And the big thing is that with these new CRISPR+ methods, you really don't have the space for an AAV vector to be able to use them. And so people have suggested that you're going to deliver 2 or 3 AAV vectors to the same cell. From our perspective, people have tried this with gene therapy for some time, and it's hard enough to get one AAV to deliver. So what we're showing here is a bunch of different of these CRISPR plus methods on the left, and then the size limitation of an AAV shown here in red across the size of the cargo on the bottom. And what you can see is, apart from maybe some very simple base editors, you're not able to package something like SA-Cas9, which is the original compact Cas9, into a single AAV. And so that's where people have proposed they use multiple ones. Here with Cas-Phi, again, our kind of medium-sized system, you're able to comfortably package these and have room to spare for most of them. And these bottom 2, RetroCas being a gene writing system, and then CRISPR-off being a permanent epigenetic silencing, you're able to fit you're not able to fit these, but with our smaller systems, you are able to package those into a single AAV. So really opening up these CRISPR plus methods, not just as tools for cell editing, but allowing them to move in vivo, which again is the real promise of gene editing. All right, so now for the last couple minutes, I'll switch over to disease detection. So apologies for the sharp transition, but I think this is an exciting area that probably less people have thought about, and I think is something that there's huge potential, despite diagnostics not always getting as much attention as therapeutics for CRISPR. So really, the goal of diagnostics using CRISPR is to drive drive the accuracy of a centralized testing lab. So something like you'd send out to a centralized lab and get a result, which are often qPCR-based assays, and being able to move that into a decentralized setting. So having that accuracy, but in a much simpler and much more affordable format. And this is going way back to our work in the Doudna lab. But one of the interesting observations, again, just thinking about the natural diversity of these CRISPR systems, is when we started to look at Cas12a in this case versus Cas9, and when it bound to its activator, so you can think about this as a genetic target. One thing we saw, which was very surprising and strange at the time, was that this Cas12a enzyme rapidly degrades the target and the trans target, so the DNA that's not being bound by the CRISPR system, and rapidly degrades it to nothing essentially, versus Cas9, which doesn't do anything to the single-stranded target. So originally, of course, we thought this was a contaminant or something like that. Digging into it, we were able to confirm that this was the case. And we quickly started to adapt that for disease detection. So what we do here is we replace that single-stranded DNA with a reporter that has a fluorophore and a quencher. And once the CRISPR system gets activated, so it binds to its target, it's able to cleave that, and it sends out kind of this molecular flare that we can detect. Of course, when we made this discovery, shortly after, we started to enter into the COVID pandemic, which was, I think, a time where we felt like we could contribute and start to develop this assay for COVID. And so this is just a highlight of some of the different milestones that we made throughout the pandemic, where we actually had one of the first EUAs, so the FDA's emergency use authorization to actually deploy this for a high throughput COVID test. I think this is the cool thing about this. The EUA that we had is, there's this checklist at the submission forms that you have for the FDA, and they actually mostly, ELISA, PCR, these technologies that are decades old. And during the pandemic, with our submissions, they actually had to add another checkbox on there that was a CRISPR-based checkbox. So I think really showing that you can have a new technology in a relatively stagnant field for diagnostics and have it move relatively quickly. So this is highlighting some of the data that we generated throughout the pandemic. Here, showing analytical sensitivity against a PCR instrument that's about the size of a room versus being able to do this even on a benchtop, and you can see comparable specificity and sensitivity to those gold standard PCR assays. I think another really cool thing highlighting the specificity of this, which I think is really the power of these CRISPR enzymes, is that as new variants were coming out, we were able to make new assays and just really quickly reprogram it in a matter of weeks for new targets that were emerging, and get high concordance, perfect concordance in this case, with the whole genome sequencing benchmark assays. So differentiating things like Omicron from Delta and the original variants, just by using a few different guides to target different areas of diversity. So again, long-term, the vision, it doesn't necessarily need to be at home, but even in a clinic or in your physician's office, being able to drive diagnostics into this decentralized setting where you can actually have the molecular information that you need. I think also a lot of overlap with the indications that we're looking at from therapeutic targets, where you can actually stratify patients and identify the right therapy for the right patients using this technology as well, without having to send it out for weeks. So I think with that, I'm probably at time. Thank this outdated list of the Mammoth team. We're about 190 people at this point, and yeah, of course they did all the work, and very, very grateful for the team that we have and the people that work with us. Yeah.
Thank you very much.
Well, thank you everyone for your attention.
So I think we should clap our hand one more time for Lucas. Excellent presentation. Thank you, Lucas.